In this video, you will be informed about the definition of a gene mutation, the types of gene mutation.
Assalamu alaikum. A gene mutation is a change in the nucleotide sequence of the DNA. It can be harmful, such as mutation leading to cancer or some diseases like sickle cell anemia. As you see here, in sickle cell anemia, the red blood cells, which are typically round and flexible, become abnormally shaped like a crescent or sickle. Also, mutation can be beneficial, such as mutation protecting from some diseases like atherosclerosis. In atherosclerosis disease, the arteries become narrowed and hardened due to the accumulation of plaque in their inner walls. This blocks the blood circulation.
A gene mutation can be substitution, deletion, or insertion.
A mutation is substitution when one nucleotide is replaced by another. Here we give you a part of the non-transcribed DNA sequence of a normal gene and that of a mutant gene, so subjected to mutation. Let's see the mutation. So, as you see, A is replaced by T. So this mutation is substitution. To determine the amino acid sequence for each gene, normal and mutant, we have to first to write the mRNA sequence and second to refer to the genetic code table in order to write the amino acid sequence. But as we give you the non-transcribed DNA sequence, so in this case the mRNA sequence will be identical to the DNA sequence, but instead of T there is U. So here we have T is replaced by U, and here we have T is replaced by U and T is replaced by U. By using the genetic code table, we can write the amino acid sequence encoded by each gene. The second amino acid sequence here is GLU. As you see here in the amino acid sequence encoded by the mutant gene, it is replaced by VAR. A mutation by substitution can be silent. What does this mean? This means that there is a change in the DNA sequence, but no change in the amino acid sequence. Let's watch the animation. So here, G is replaced by A. But as you see here, we have the two amino acid sequences which are identical. GLU remains GLU. So the mutation passed unnoticed. A mutation by substitution can be a nonsense mutation. What does this mean? This means that the change in the DNA sequence leads to a stop codon in the mRNA. So, let's see this animation. Here, G is replaced by T. So, in the mRNA, we'll obtain UAG, which is a stop codon. In this case, the synthesis of the amino acid sequence will be interrupted. It results in an incomplete and short protein. The second type of gene mutation is mutation by deletion. In mutation by deletion, one or many nucleotides are removed. If we talk about point mutation, so one nucleotide will be removed or deleted. In this example, A is removed, causing a frame shift within the sequence. And as you know, a triplet of nucleotide is read as a codon. If a single nucleotide is deleted, so the triplet grouping will be disrupted. And as a result, all the amino acids situated after the site of mutation will be or may be changed. As you see here, we have after the site of a mutation we have threonine, and here we have proline and arginine. The third type of gene mutation is mutation by insertion. Mutation by insertion occurs when one nucleotide is added. In this example, G is added to the sequence causing also a frame shift like in deletion mutation. And also, like deletion mutation, all the amino acid obtained after the site of a mutation will be or may be changed. Here we have threonine and threonine, and here we have asp and his. Finally, in both mutations, deletion or insertion, the protein obtained will be non-functional in general.
The gene mutation can be substitution when one nucleotide is replaced by another, or deletion when one nucleotide is removed, or insertion when one nucleotide is added or inserted.